Okay, so today we are going to go through our process of transcription and translation. So we went over transcription, translation just a little bit last time. We went over where they occur and what is forming in each. So when we have transcription, we're going from DNA to RNA. And when we have translation, we're going from RNA to a protein. So we've talked about proteins in the past. We've talked about DNA in the past. We really haven't talked too much about, RNA yet. Now, RNA, just like, especially the ones in our bodies, just like DNA, it's made of nucleotides. Difference, so one difference between DNA and RNA. DNA has the sugar deoxyribose. RNA has the sugar ribose, so a little bit different. The other difference there is one of our bases is different in RNA compared to DNA. So DNA has thymine. If you remember, DNA had thymine. RNA does not have thymine. Instead, RNA has a different base called uracil. So uracil just takes the place of thymine. Let me repeat that. Uracil just takes the place of thymine. That's it. So if you're starting off with DNA and you have an A in DNA, usually if it's another DNA, the A would pair to a T. Instead, if we're going to RNA, the A is going to pair with the U. One other difference, so here's three differences right here. We have one, the ribose sugar, two, the thymine. Another difference, RNA is going to be single-stranded instead of double-stranded like DNA. It's a little bit shorter as well. You can use that as a difference. All right, so there's actually four differences between DNA and RNA. So if we want to do base pairing, I put the DNA bases in blue and the RNA bases in red just to make it easy on you guys so you can see the difference. If we have A in DNA, that's going to go to U. If we have G in DNA, it's going to go to C. If we have C, it's going to go to G. So nothing changes with those two. If we have a T in DNA, it still goes to A. So again, nothing changes. The only one where anything changes is the A going to the U. So if we're going from DNA to RNA, it goes A to U. You could also go RNA to DNA if you wanted to. And in that case, we would have the U going to the A of DNA, okay? All right, so three different types of RNA we're going to go over that are in our cells. Our first one, which is going to be the main one we're going to use, is called mRNA. This is the RNA that is made by the DNA to leave the nucleus. Again, this is the RNA that is made from the DNA and it leaves the nucleus, all right? What the mRNA does is it carries the message of DNA. Our goal is to make a protein. It carries that protein message. Our RNA, that just makes up the ribosomes, whether they're free ribosomes or ribosomes on the rough ER. And our tRNA, the last one, the tRNA is a type of RNA that's going to bring the amino acids, bring in our subunit of the protein to make the protein. And I'll show you guys different things with that. So here is everything we're going to be going over. We have our transcription, where our mRNA is going to leave the nucleus. The mRNA is going to come to the ribosome, which is our RNA. And this tRNA, this guy over here, he is going to attach to our mRNA. And on the other side, he's going to have his amino acid to build the protein up. So if you remember, amino acids are the subunits of protein. So a bunch of amino acids together make a protein, okay? So that's our whole process, guys. So the tRNA is going to bond to the mRNA. And again, I'll show you everything here. Now, when we are making RNA, specifically mRNA, we are going to use our DNA as a template. And what we're going to do is we are going to use that DNA in order to make our mRNA. And if you remember, last time we made DNA from DNA, we used a guy called DNA polymerase to attach our nucleotides. To make RNA, we're going to do the same thing. We're going to attach nucleotides. But we're making RNA, not DNA, so we call it RNA polymerase instead of DNA polymerase. Now, just to go over something that's, kind of going on now, some viruses actually are made of RNA. RNA polymerase, which that's how viruses are going to replicate their DNA, RNA polymerase actually doesn't have a proofreading thing on the back like DNA does. So if you remember, DNA polymerase had that nice proofreading thing, so if there was a mutation, we could fix it. RNA does not. So if there's a mutation, kind of lets it go. This means viruses that are made of RNA actually have a higher mutation rate compared to the DNA ones. All right, so in the end, RNA polymerase is going to do its job. It's going to add nucleotides, start to build up our mRNA molecule, and at the end, it's going to reach a signal that'll terminate the whole thing and be like, hey guys, guess what? We're done. Okay, cool. I made my RNA now. All right. So here's the process, guys. You could see transcription. That's DNA going to RNA. Our DNA unwinds, just like it did last time. As it unwinds, we are going to have this mRNA being made off of this DNA. And I'm going to show you guys the base pairs here in a minute. And as we have our polymerase here, this one specifically is RNA polymerase. As we have that polymerase, we're going to add nucleotides to it. And so it's going to grow bigger and bigger and bigger. All right. Here's another picture of it. And you can see the pink there is our mRNA growing. So as we unwind the DNA in a certain area, we're going to add more RNA nucleotides to it. Okay, so let me give you guys a quick example of this. So I'm going to go to the complementary DNA. Sometimes it'll ask you to, sometimes it won't. So let's go to the complementary first. And I'm just going to write it right above the complementary DNA, guys, but just so you know, I am making it. I'm going to make it in red. So this is DNA going to DNA. A goes to T, C goes to G, C goes to G, G goes to C, A goes to T, A, C, and G. Nothing too crazy there. All right. Let me white some of this out a little bit here, just so you guys can see a little better. Okay, good enough. I just don't want the test tube and all that stuff getting in the way. That's all. Good enough. All right, so let's do our next one here. So our next one, let me move myself out of the way here. I don't know what happened there, but let me move myself back. Okay, next one, guys, we are going to make some mRNA. Now, it might tell you to go off the original. It might tell you to go off the complementary. I'm going to go off the complementary here. So the T would go to A. The G would go to C. So if you guys notice, this strand I'm making is going to be pretty much the same as my original strand with one exception. And that one exception is wherever there was a T up here, they changed to a U down there. Everything else is the same. This is my mRNA. This is the part that's going to go to the ribosome. When it's on the ribosome, the tRNA is going to fit perfectly into the mRNA. And that tRNA on the other side of it, it's going to deliver the amino acid. So we pretty much do the same thing. So A would go to, and this is RNA again, A would go to U, G, G, C, A goes to U, A, let's see if I'm still there. Last ones, guys. Sorry, I don't know what's going on here with the internet. G would go to C, and then the C would go to G. All right, that's all you do for this to do your tRNA, your mRNA. Just make sure your only difference is when you're going to RNA, you want to make sure you're using U's and not T's. Okay. All right. So when we are doing our mRNA, we are going to use 3 bases, so like an A, a U, and a U, in order to get the amino acid. And I put some stuff on Canvas for you guys so you can see what I mean by this, and we'll go over it in a little bit. But we're going to have things that start a sequence, and we're going to have things that stop a sequence. For mRNA, we are going to have start codons and stop codons, and all it is 3 base pairs at the beginning and 3 base pairs at the end. That's all these mean. So anytime you guys see codons, codons are talking about mRNA. So now we're going to make a protein. This is going from our mRNA to the amino acids. We have 20 different amino acids we can choose from. All right. So we're going to use our mRNA, and we're going to go to these amino acids. So as I did in that example, my mRNA and my tRNA, they're going to join together. The mRNA, let me label this on here. The mRNA is going to be the codon. That's just the vocab word for it. The tRNA is our anticodon. And our codon and our anticodon are going to bind together. The anticodon, the tRNA, this is the one that is going to carry the amino acids. All right, so the tRNA is going to carry the amino acids. Okay, so in this process, guys, we're going to have it elongating. We're going to have an amino acid chain just getting longer and longer and longer. And then once it reaches the end, we're going to have one big protein being made. That's these 2 vocab words for you here. After we're done, the tRNA drops off the amino acid. After it drops it off, it doesn't have that amino acid anymore, so it has to go find a new one. And here's the whole process here. The tRNA is this little spaceship-looking guy here. The amino acid, that's attached to it, and we have 20 different ones. And if you remember from chapter 3, why we have 20 different ones is we have 20 different R groups we can choose from. So the tRNA is going to come, it's going to bind to this codon, which is the mRNA here, and when it has a correct fit, we are going to let that protein, that amino acid, come off. All right? So here are all of your amino acids. And I'm going to teach you guys how to use this chart, okay? So this is one version. This is the other version. I've seen this on the keystone. I've also seen this on the keystone. All right? So I want to make sure you guys can use both charts. This one is easier. This one, a little bit more difficult. If we wanted to go to Canvas here. So for you guys, I have our codon charts here. One of them will be right here. I gave you guys a little bit of a nicer one there than the one I have in the PowerPoint. And the other one right here, that's the circular one. Tried to make it colorful for you guys. All right, so you have access to both of them. Let me show you guys how to use one. So let's say I have a sequence of, I'm going to make this a little bit smaller. Let's say I have a sequence of mRNA. So we're going to use these charts always, always, always for mRNA. That's the only thing we're using these charts for. So let's say the mRNA is A. That is not a good color to use right now. Let's get rid of that. And say it's A, U, G. And when you're writing your mRNA, guys, it always helps to put it in threes. So AUG, let's do GGG, U, G, C. Let's do U, G, A. Here's how we use this chart. Over here, this is going to be our first letter. Up top, this is our second letter. And over here on the right side, this is our third letter. So let's look at our first triplet. Let's look at our first codon. Again, this is the mRNA. So if we have AUG, start off with the first letter. First letter is A. So here's our A. We know it's going to be somewhere in this whole mess right here. It's going to be one of these. So we eliminated all the other ones. So that's our first one, A. Second letter, U. All right, now we know it's going to be one of these ones right here. All right. Third letter. Our third letter is a G. Oops. There we go. Okay. So our third letter was a G. And I don't know why I keep not disappearing, but whatever. On the last letter, AUG, we have our A, we have our U. Now the G is the fourth letter down on the third. So that tells us our amino acid, this is an amino acid chart, is methionine. So M-E-T, and I'm just abbreviating it. Okay? Second one. Again, same thing. GGG. First letter, G. Second letter, G. Last letter, oops, we're cut off there. Let's see what it is. All right, it's the same as all of them. So that last one is glycine, G-L-Y. UGC, U, first one, G, C, cysteine. I'm just going to abbreviate CYS. And our last one, UGA. U, G, A. It says stop. That is a stop codon. That means we are at the end of our chain. Now, these are going to be very big, guys. They're not just going to be 4 amino acids long. They're going to be hundreds, but we're never going to do one that's like 100 long. All right? But that's how you do that one there. Let me go to the next one here. So a little bit of a different chart, same premise. So let's say we have A, C, G. What you do is you start at the bullseye, right in the middle. First circle is your first letter. So the first letter is A, I'm going here. Second letter is C. Okay, I'm going to the C. Last letter is G. G. It goes to threonine. All right. And that one, all 4 of them give the same amino acid, which is kind of nice. Let's do GAU. Start off in the middle. You go to G, you go to A, you go to U. It's aspartic acid. All right. That's pretty much it, guys. That's all we're doing for this. I'm going to do more practice with you guys on this tomorrow, give you some example problems, and then we will go from there. All right, guys. Have a good rest of your day. I'll see you tomorrow.